Welcome to this introduction to the PAIA assay technology. In the next 5 minutes we will show you how to successfully run our bead-based immunoassays that allow you to rapidly quantify antibodies and proteins. These are the 4 different steps of the PAIA assay procedure, which are essentially the same for each PAIA assay. First, all assay components are added into the wells of the microplate, and this mixture is incubated on a shaker. Once this is done, the beads are allowed to settle to the well bottom by sedimentation, which then allows to read the microplate on the fluorescence reader or microscope. Our assay kits contain the special PAIA microplate and a ready-to-use buffer with a fluorescence marker. In the first step of the assay, the buffer is dispensed onto the dried-in beads in the microplate. Typically around 50 microliters of buffer is added into each of the wells. For this demonstration we use 6 rows of the microplate, 3 for calibration standards and 3 for the samples. The remaining wells can be used later in a different experiment. If you want to do this, you should keep the microplate and the buffer in a fridge at 4 degrees. Now we are adding the calibration standards to the first 3 rows. We recommend to prepare at least 10 calibration standards for the generation of a reliable calibration curve. For optimal results, it is advisable to prepare the standards in the same matrix as the samples. The next step is the addition of samples into the wells in rows 4 to 6. The samples can be crude, for example cell culture supernatants, and used without dilution. Ideally, the samples are already in a 384 well format as shown here. All pipetting steps in a PAIA assay can be performed by a liquid handling system, which facilitates the process, especially if your samples have to be transferred from other microplate formats than 384 wells into the plate. Now the plate is ready to incubate. You may seal the plate prior to incubation, but this is not necessary if you have checked that the samples do not spill over during incubation. It is important that the shaking is efficient. Depending on the shaker model, the shaking speed should be set between 1400 and 1800 RPM. Once the shaking is finished, allow the beads to settle for 10 minutes on the bench or on the shaker. Afterwards, the plate is ready for measurement. The readout of the plate is done by fluorescence detection from the bottom of the plate. Please refer to the kit manual for the correct wavelength settings and plate dimensions. You can also use an automated fluorescence microscope with very similar readout speed and assay performance. The measurement takes not more than 3 minutes for the entire plate. Here you can see the different images the microscope takes in the center of each well. You can quickly identify the ascending calibration curves in the first 3 rows and directly spot the samples with high and low concentrations in the rows 4 to 6. The data analysis can be performed either by the software of your reader or by using the PAIA evaluation tool. It is an easy-to-use software package that was specifically developed for PAIA assays. It generates calibration curves, heat maps, calculates unknowns, and creates reports. If you want to learn more about the PAIA technology, please check our website, paiabio.com. Thank you.